Welcome to AP Biology. Today's presentation is going to be on the genetic code and mutations. So our genetic code is made up of 4 letters, and each word in this genetic code is 3 letters long. We call these words codons. And they discovered this quite a while ago, 60, 70 years ago. And the way they figured it out is they knew each letter couldn't be a word, because then there'd only be 4 possible words, and that wouldn't be enough to code for all the amino acids. And they knew it couldn't be 2 letters, because that would be 4 possibilities times 4 possibilities, so that would only be 16 possibilities. And we have 20 amino acids in our body. So it seemed like 3 letters was a little redundant, because now there's 64 possibilities and we only have 20 amino acids. But it turns out the code's a little redundant. And so when you look at the actual code, which we'll look at in a minute, there's a lot of different codons that code for the same amino acids. So basically what goes on is we have our DNA molecule, which codes for a messenger RNA molecule. And these messenger RNA molecules are going to code for different amino acids. And then we're going to put those amino acids together, and how the side chains on those amino acids interact. You're going to form different shapes, and those different shapes have different functions. And so the shape of a protein is going to determine its function. And so you have a codon, say like AUG here, which is going to code for methionine, and CGU, which is going to code for arginine. And you go right down the line, and every 3 letters codes for a particular amino acid. Now, I said that there's a little redundancy in this code. So this is the kind of decoder for the code. And so the way you look at this is like C, C, C. So this C puts it in here. This C puts it in this column. And then this C puts it as proline. Okay? If I had U, U, G, U would put it in this box. This U would put it in the column. The G would make it leucine. Okay? Every single one of our proteins starts with the start codon, which is methionine, AUG. So you can see even this one we had here as our pretend one started with AUG. And all of our proteins are going to stop with a stop codon. Okay, and there's a few different stop codons, and they stop the process of translation. So the side chains to the amino acids are critical to the shape of the protein. So all amino acids are pretty much the same. They have a amino group, NH2, and then a hydrogen, and then a carboxylic acid group, and then they have a side chain. And the side chains vary quite a bit. You can see here's a few examples of side chains. And so each one of these side chains has some variety to it. And all these side chains are going to interact with each other in different ways. And so if you change an amino acid, you're going to change the side chain and how those side chains interact. And because of that, you end up changing the shape of the protein. Well, the shape of a protein is critical to its function. And so if you change the shape of a protein, you're going to mess up the active site of an enzyme, for example, and then it's no longer going to speed up a reaction that it's supposed to speed up. So what do we call these mistakes? They're called mutations. And if you have a mutation in your DNA, or if you have a mutation in in the transcription process, okay, those can lead to mistakes in the making of the protein. and can lead to the wrong amino acid being put into a protein, which can change the shape of the protein, which messes everything up. So there's a variety of different types of mutations. Some mutations are worse than others, and we'll look at a few right now. So we have one mutation, it's called a point silent mutation. So in this particular situation, we change the letter in the code, but it doesn't change the amino acid that's coded for. So if you think about the code, I could change a CCU to a CCC or a CCA or a CCG, and it's still going to code for proline. Those are what we would call silent mutations. They're changes, but they really don't change anything. And you can see there's a lot of redundancy in this code. Leucine, 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 valine, 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 alanine, 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 threonine, threonine, blah, 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 proline, serine. There's a lot of redundancy in this code. So a lot of times if you have a change in that third amino acid, it doesn't even change anything. And we call those silent mutations. They're changes, but they don't affect the protein at all. Another type of point mutation is what we call a missense mutation. So we're going to have a change, and it's going to cause the amino acid in that one spot to change, but that's it. Now, sometimes when you change an amino acid in one spot, it ruins it. Sometimes when you change an amino acid in one spot, it might make things better on rare occasion. And sometimes when you change an amino acid in one spot, it's what we call neutral. Most of the time, it's going to ruin the effectiveness of the protein. But sometimes it creates something. All of the new variations that we find in nature that are unique and new and have not been seen before, almost all of those are caused by point mutations. So point mutations are what allow evolution to be successful. Then you have point nonsense mutations. Those are bad. Because basically what you're going to do is you're going to replace the codon with a stop codon. So your protein only gets partially made, and partially made proteins are non-functional. And so there are point nonsense mutations that are pretty serious genetic diseases. You have a frame shift. Frame shifts are super bad too, because whether you're inserting an extra letter or deleting a letter, what you do is you shift the whole reading frame one way or the other. And basically, so from that point of the mutation on, nothing is read properly. So every amino acid gets changed. You will always get a completely non-functional protein, and it's pretty much going to be lethal. So, for example, if I take the fat cat ate the rat and I throw an R in there, now I have the fat car tat F era T. Okay, and all I did was I put an R, an extra R inserted where the T was supposed to be. So if I slide this over, and I have ate the rat still, but everything's messed up because it's all pushed one letter this way. Same thing here. If I take the A out from fat, now it's the fitic eta tet her rat. Well, that doesn't make any sense. Just like in the DNA, you're not going to make sense. You're going to have all the wrong amino acids, and you're going to make a non-functional protein. Duplications means we're going to duplicate a sequence. Those sometimes can be useful. So like the fat cat ate the rat doesn't really change the meaning. And so sometimes you can get a functional protein from that. In inversions, sometimes you can get a functional protein from an inversion. Not very often, but sometimes duplications and inversions can lead to functional proteins. Sometimes point missense mutations can lead to functional proteins. But remember, most of the time when you have a mutation, it's going to mess things up and it's not going to make things better.